Hi class, in this recording we're going to dive in and start talking about bone tissues, chapter 7 material from your textbook. As we look at bone tissue, bones and our teeth are very durable. They're very tough. Oftentimes these are only things left after the rest of the soft tissues have decayed and decomposed. Our living skeleton, though, is very dynamic. There is lots of tissue change within our skeleton. There's lots of cells, and these cells have lots of nerves, or the tissues have lots of nerves, I should say, and there's also many blood vessels perforating our skeletons. Our skeleton is going to be continually remodeling itself, which is a nice way of saying, or another way of saying that our skeleton is constantly reforming and rebuilding itself while it interacts with the other parts of our body. For those of you who are interested in focusing specifically on bones, oste is a root word here that we're going to see a lot. That means bone. Ology means study of. Osteology is the study of bones. Now, as we're looking at our skeletal system is going to be made up of bones, obviously, our skeleton. It also is going to be made of cartilage and ligaments associated with those bones. Cartilage in our bodies is the forerunner, the precursor of most of the bones in our body, particularly the long bones of our bodies. And when we talk about this process of bone formation, endochondral formation, ossification, most of our bones start off as a cartilage framework that then gets ossified or turned into a bone. To review, and hopefully this is just tacit knowledge for you, a ligament is a bone-to-bone connection at joints or arthroses. A tendon is a bone-to-muscle connection that will attach muscles to bones. Both ligaments and tendons, in particular tendons, and the stress associated with the tendons influence the shape and direction of bone growth throughout our lifetimes. If you're looking at an individual, you can look at the bony attachment points of their tendons and look at the size of those bony attachment points to infer the muscular strength of that individual. Bigger attachment points are for bigger muscles. As we look at the functions of our skeleton, they are numerous. For instance, our skeleton supports our bodies. Our arms and legs and vertebra support our body so we can stand upright. The jaw bones support our teeth. We're also going to have some bones of the abdominal pelvic cavity supporting our overall viscera. We also have protection. In particular, think of the bones of the cranial vault, those flat bones of the skull. They protect the brain. The vertebra protect the spinal cord. Our thoracic cavity protects our heart and lungs. We have a lot of squishy organs that are protected by our bones. Our bones aid us in movement. They serve as attachment points for skeletal muscle tissue, and when that skeletal muscle tissue when that skeletal muscle tissue contracts, that skeletal muscle tissue is going to apply force to the bone and cause the bone to move. Our bones aid us with electrolyte balance. To review from chapter 2 material, an electrolyte is a dissolved ion in water that conducts electricity. So as we look at calcium ions and phosphate ions, these are two important electrolytes within our bodies, or minerals within our bodies, that we're going to need to help maintain homeostasis for, and our skeleton, the bones of our skeleton, help us to maintain calcium and phosphate homeostasis. We also regulate our acid-base balance with our skeleton. In particular, we have buffers in our blood that are based on phosphates, and these phosphate buffers are going to resist changes in pH. We also have carbonate buffers as well. Those are not talked about as frequently in conjunction with the skeletal system. Typically, we focus on the carbonate buffers when we get to the respiratory system in A&P2. We also are going to have blood formation with our bones. In particular, we have a tissue in our bones called red bone marrow, and red bone marrow has our precursor cells for our blood. Most of our blood is made in the red bone marrow of our skeleton. As we look at our bones, bones can be referred to as osseous tissues. Osseous tissue is a category of connective tissue that we've already talked about when we covered histology. Notably, this is going to be solid at body temperature. And by solid, I mean stiff solids. There's very, very little flexibility to the bones. The matrix of the bone, the extracellular portion of bone tissue, is going to be hardened by calcium phosphate and a variety of other minerals associated with calcium and phosphate. There are multiple kinds of osseous tissue. We have spongy bone and compact bone being the two notable kinds of osseous tissue. The process of making a bone or hardening a bone, I should say, is referred to as calcification or mineralization of the bone. And this is when we add more calcium or more minerals to that extracellular matrix to make the extracellular matrix of the bone stiffer or harder. When we look at an individual bone, we can consider them an organ because there are multiple tissue types working together for a common purpose. We have that osseous tissue, we have the marrow, the cartilage, we have some adipose, there's some nervous and fibrous connective tissues all associated with an individual bone. So an individual bone can be thought of as a tissue type if we have dense bone or spongy bone, or a bone can be thought of an organ if you think of the individual bone as a whole structure and all the individual tissues associated with that individual bone. We can categorize bones based on their general shape. Long bones are one of the easiest to recognize. As a rule of thumb, long bones are going to be longer than they are wide. I like to say twice as long, at least twice as long as they are wide. Our long bones are primarily going to be rigid levers in our bodies to aid us with movement. They typically are going to be associated with monoaxial or biaxial joints. The notable exceptions are going to be the head of the femur and head of the humerus, which are associated with the shoulder and the hip as a multi-axial joint. Our short bones are approximately equal in length and width. Classic examples of short bones include the tarsals and the carpal bones of the feet and hands. And these short bones can glide across each other glide across each other's, excuse me, I'm having some grammar issues right now. They can glide across each other in multiple directions. So there's multiple planes of movement. Typically, short bones are going to be associated with biaxial joints. As we look at our flat bones are thin bones that have curved plates. They typically are going to be associated with protecting soft organs. The flat bones of the cranial vault, the temporal, the frontal, the parietal bones are oftentimes thought of. Other flat bones include the scapula or the sternum of the thoracic cavity or thoracic cage, even though the scapula is typically referred to as part of the upper appendage. We also have irregular bones, and this is the miscellaneous category. These are bones that just have wonky shapes. Bones of the face are oftentimes referred to as irregular bones. For example, here's the sphenoid, one of the bones of our face. Bones of the vertebral column are also irregular bones. It's hard to categorize the bones based on these physical shapes, so we just say they're all irregular, the miscellaneous category. When we look at generic bone or generic long bone, all these bones are going to have compact or dense bone tissue on the outer perimeter. Here we can see a femur that's been sliced along a frontal or coronal plane, and you can see how there's some compact bone around the entire perimeter. And then in the middle of the epiphyses, we have a lot of spongy bone tissue. Spongy bone tissue is my preferred name. Miscellaneous is an older school name. A really old school name is the trabecular bone. They all mean the same thing. When we look at the spongy bone tissue, it's loosely organized and has lots of little hollow spaces in it. And these little hollow spaces are going to be where we store most of our red bone marrow. We typically find spongy bone tissue at the ends of long bones or in the middle of nearly every other bone of our body. Spongy bone tissue should never be on the surface of the bone. It should always be deep to compact bone tissue. That compact bone tissue is much more durable than spongy bone and protects spongy bone from damage. When we look at the skeleton, the mass of our skeleton is approximately 75% compact bone and 25% spongy bone tissue. So most of the mass of our skeleton is going to be within that that compact bone tissue, because of the lack of air pockets or hollow spaces within that compact bone tissue. As we look at a generic long bone, our generic long bone can be broken up into two primary regions. We have the ends, called the epiphyses, and then we have that middle portion called the diaphysis. And we can have a proximal epiphyse and a distal epiphyse based on the specific appendage you're looking at. The epiphyses are typically going to be the anchoring points for ligaments and tendons, and the diaphysis gives physical length and leverage to the long bone. And as we look at the middle of the diaphysis of a long bone, there's the marrow cavity or medullary cavity in the middle. When we are younger, this marrow or medullary cavity is oftentimes filled with red bone marrow, and as we age, the marrow cavity fills with yellow bone marrow or adipose tissue. To separate the diaphysis from an epiphyses, we have an epiphyseal plate or epiphyseal line. When we are still growing taller, so before our when we're young children and young adults, we're still growing taller, we call it the epiphyseal plate, oftentimes referred to as the growth plate. And it's a line of hyaline cartilage that separates the epiphyses from the diaphysis of a long bone. Once we stop growing taller, the hyaline cartilage becomes fully ossified and now is going to be called the epiphyseal line. And it's just a line of solid, dense bone tissue separating the spongy bone tissue of the diaphysis from the spongy bone tissue of the epiphyses. This epiphyseal line will fade as we age, and in aged individuals, the epiphyseal line is very difficult to see, and oftentimes you'll see nothing but spongy bone tissue. And to differentiate the epiphyses from the diaphysis, you oftentimes need to look at the trabeculae orientation within the spongy bone tissue, or the bone fiber orientation within that spongy bone tissue, in order to differentiate the epiphyses from diaphyses. As we look at the ends of a long bone, the proximal and distal ends of a long bone that are in a joint are going to be covered in hyaline cartilage. This hyaline cartilage is referred to as articular cartilage. Articular cartilage is a special kind of hyaline cartilage that reduces friction primarily. It also provides some shock absorption, but primarily it helps to reduce friction within a joint. We also have many small holes within the bone known as nutrient foramen. And these nutrient foramen, or foramina, depending on plural versus singular, are going to be little holes in the bone that let blood vessels and nerves penetrate the bone. We want to emphasize blood vessels because blood vessels are going to be the means of delivering nutrients and removing waste products from our bones. Our long bones our bones also have some membranes associated with them on the inside and the outside. On the inside of the marrow cavity, we have a membrane known as the endosteum, or endosteum. Endo meaning in, osteum referring to bone. So this is a membrane that lines the marrow cavity. It's going to be right on the inside. I'm over here in the lower right-hand side of your screen in green. I'm outlining where the endosteum would be. And that endosteum is going to have some osseous cells associated with bone remodeling in them. We also have the periosteum all around the outer perimeter of the bone. Peri referring to perimeter, osse referring to bone. The periosteum is a membrane that's folded back right here in this figure that is going to wrap around the outer perimeter of the bone. It's made up of a lot of collagen fibers. Some of those collagen fibers will penetrate the dense bone tissue. Those are referred to as the perforating fibers, and they are going to anchor the periosteum to the compact or dense bone tissue around the perimeter. And some of those fibers are going to be continuous with the tendons and ligaments associated with the bones. On that inner layer of the periosteum, we have some osteogenic cells. Oste, again, referring to bone. Genic is a root word that means genesis or creation. So we have some bone creating or bone forming cells on the inner layer of the periosteum that allow for our bones to grow wider and also allow for our bones to heal after we break them, after we have a fracture occur. As we look at flat bones, so we're transitioning from long bones to flat bones. Flat bones are going to have a sandwich-like construction. There'll be compact bone on top and bottom, and then in the middle of our sandwich, instead of a cream filling, we have spongy bone tissue. So we have the two layers of compact bone that provide lots of structural support, and then that spongy middle layer, often of spongy bone tissue, is referred to as the diplo, and that's going to allow for shock absorption. A lot of new bicycle helmets that are being made are actually mimicking the diplo of the flat bones. So I'm thinking of some new helmets that I just saw hit the market this year. So instead of having individual pads on the inside of the helmet, there's kind of a honeycomb matrix on the inside of these bicycle helmets that resembles the spongy bone tissue of the plates of our skull. It's worth emphasizing that the marrow spaces in these little tiny little cavities in the spongy bone tissue are all going to be lined with endosteum. And then the inner and outer, or I should say the superficial and deep, flat, dense bone tissues are going to have periosteum lining them as well. So we're still going to have those membranes of the long bone, but they're going to be oriented in slightly different fashions, particularly the endosteum, because there is no marrow cavity of our flat bones. That's all we have for this recording. If you have any questions or comments, please feel free to post them in the class discussion board or to shoot me an email. And as always, class, happy studies.